This speaks to embryogenesis, which I think is one of the coolest things in the universe. In fact, you acknowledge its coolness in Ingressing Minds paper, writing, quote, most of the big questions of philosophy are raised by the process of embryogenesis, right in front of our eyes. A single cell multiplies and self-assembles into a complex organism, with order on every scale of organization and adaptive behavior. Each of us takes the same journey across the Cartesian cut, starting off as a quiescent human oocyte, a little blob thought to be well described by chemistry and physics. Gradually, it undergoes metamorphosis and eventually becomes a mature human with hopes, dreams, and a self-reflective metacognition that can enable it to describe itself as not a machine. It's more than its brain, body, and underlying molecular mechanisms, and so on. What in all of our discussion can we say as the clear intuition how it's possible to take a leap from a single cell to a fully functioning organism full of dreams and hopes and friends and love and all that kind of stuff? In everything we've been talking about, which has been a little bit technical, like how do we understand? Because that's one of the most magical things the universe is able to create, perhaps the most magical. From simple physics and chemistry, create this. Us two talking about ourselves.
I think we have to keep in mind that physics and chemistry are not real things. They are lenses that we put on the world, that they are perspectives where we say we are, for the time being, for the duration of this chemistry class or career or whatever, we are going to put aside all the other levels and we're going to focus on this one level. And that what is fundamentally going on during that process is an amazing positive feedback loop of collective intelligence for the interface. It's the physical interface is scaling its the cognitive light cone that it can support. So it's going from a molecular network. The molecular network can already do things like Pavlovian conditioning. You don't start with zero. When you have a simple molecular network, you are already hosting some patterns from the Platonic space that look like Pavlovian conditioning. You've already got that starting out. That's just a molecular network. Then you become a cell, and then you're many cells, and now you're navigating anatomical morphospace, and you're hosting all kinds of other patterns. And eventually, you and I think again, I think there's, and this is like what, all the stuff that we're trying to work out now. There's a consistent feedback between the ingressions you get and the ability to have new ones, which again, I think it's this like positive feedback cycle where the more of these free gifts you pull down, they allow you physically to develop to ways where, oh, look, now we're suitable for more and higher ones. And this continuously goes and goes and goes until you're able to pull down a full human set of behavioral capacities.
What is the mechanism of such radical scaling of the cognitive cone? Is it just this kind of the same thing that you were talking about with the network of chemicals being able to learn?
I'll give you two mechanisms that we found, but again, just to be clear, these are mechanisms of the physical interface. What we haven't gotten is a mature theory of how they map onto the space. That's just beginning. But I'll tell you what the physical side of things look like. The first one has to do with stress propagation. So imagine that you got a bunch of cells, and there's a cell down here that needs to be up there. Okay, all of these cells are exactly where they need to go, so they're happy. Their stress is low. This cell, now let's imagine stress is basically a it's a physical implementation of the error function. It's basically the amount of stress is basically the delta between where you are now and where you need to be. Not necessarily in physical position. This could be in anatomical space and physiological space and in transcriptional space, whatever, right? It's just the delta from your set point. So you're stressed out, but these guys are happy. They're not moving. You can't get past them. Now imagine if what you could do is you could leak your stress, whatever your stress molecule is. And the cool thing is that evolution has actually conserved these highly. So these are all, and we're studying all of these things. They're actually highly conserved. If you start leaking your stress molecules, then all of this stuff around here is starting to get stressed out. When things starting to get stressed out, their temperature, in the not physical temperature, but in the sense of like simulated annealing or something, right? Their ability to, their plasticity goes up because they're feeling stress. They need to relieve that stress. And because all the stress molecules are the same, they don't know it's not their stress. They are equally irritated by them as if it was their own stress. So they become a little more plastic. They become ready to kind of, adopt different fates. You get up to where you're going, and then everybody's stress can drop. So notice what can happen by a very simple mechanism. Just be leaky for your own stress. My problems become your problems. Not because you're altruistic, not because you actually care about my problems. There's no mechanism for you to actually care about my problems, but just that simple mechanism means that faraway regions are now responsive to the needs of other regions, such that complex rearrangements and things like that can happen. It's alignment of everybody to the same goal through this very dumb, simple stress sharing thing.
Via leaky stress.
Leaky stress, right? So there's another one. There's another one which I call memory anonymization. So imagine your two cells, and imagine something happens to this cell, and it sends a signal over to this cell. Traditionally, you send a signal over, this cell receives it. It's very clear that it came from outside. So this cell can do many things. It could ignore it. It could believe, it could take on the information. It could just ignore it. It could reinterpret. It could do whatever. But it's very clear that came from outside. Now imagine the kind of thing that we study, which is called gap junctions. These are electrical synapses that could directly link the internal milieus of two cells. If something happens to this cell, it gets, let's say it gets poked and there's a calcium spike or something, that propagates through the gap junction here. This cell now has the same information, but this cell has no idea. Wait a minute, is that my memory or is that his memory? Because it's the same, right? It's the same components. And so what you're able to do now is to have a mind meld. You can have a mind meld between the two cells where nobody's quite sure whose memory it is. And when you share memories like this, it's harder to say that I'm separate from you. If we share the same memories, we're kind of, and I don't mean every single memories, right? So they still have some identity, but to a large extent, they have a little bit of a mind meld. And there's many complexities you can lean on top of it. But what it means is that if you have a large group of cells, they now have joint memories of what happened to us, as opposed to you know what happened to you and I know what happened to me. And that enables a higher cognitive light cone because you have greater computational capacity, you have a greater area of concern of things you want to manage. I don't just want to manage my tiny little memory states because I'm getting your memories. Now I know I got to manage this whole thing. So both of these things end up scaling the size of things you care about. And that is a major ladder for cognition is scale the degree of, the size of concern that you have.
It'd be fascinating to be able to engineer that scaling.
Yep.
Probably applicable to AI systems. How do you rapidly scale the cognitive cone?
Yeah, we have some collaborators in a company called Softmax that we're working with to do some of that stuff. In biology, that's our cancer therapeutic, which is that what you see in cancer literally is cells electrically disconnect from their neighbors. When they were part of a giant memory that was working on making a nice organ, well, now they can't remember any of that. Now they're just amoebas, and the rest of the body is just external environment. And what we found is if you then physically reconnect them to the network, you don't have to fix the DNA. You don't have to kill the cells with chemo. You can just reconnect them, and they go back to, because they're now part of this larger collective, they go back to what they were working on. Mm-hmm. And so yeah, I think we can intervene at that scale.